Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
This is Richard Jacobs with the Finding Genius Podcast. This is the Health, Medicine, and Bioscience edition. My guest today is over in India. Let me grab his bio for a second. Okay. His name is Prashanth Suravajhala. He works at a research place over in India, and we're going to be talking about exomes. He has a PhD in systems biology, Department of Biotechnology and Bioinformatics. Again, a research scientist at the Birla Institute of Scientific Research in Jaipur, India. So, Prash, thanks for coming. How are you doing?
Yes. Thank you so much, Richard. Thank you for your kind introduction. Very happy to be here. Yeah, thank you so much. Doing good. How do you do?
Good. Well, tell me about exomes. There's a lot to look at. It's a new field, growing field. What is it that you're studying about them?
Right. Yeah. So basically, come next generation sequencing, one needs to identify a lot of mutations for certain diseases. So if a disease is of subsequent interest to a research community, or if the disease is pretty rare, or if the disease is coming from a geographically specific niche area, then probably clinical exomes is a correct answer. So from next generation sequencing approaches, there are two entities. One is the whole exome sequencing, and the other one is whole transcriptome shotgun sequencing. So these two are in addition to the whole genome sequencing approaches that the next generation sequencing, or the so-called the now generation sequencing, has seen of ages. Yeah.
How do you sequence all? You're sequencing all the exosomes that a cell puts out. Is that what you're saying? That's possible?
I beg your pardon, Richard. It's not exosomes, it's exomes. Exomes by meaning coding only the exonic chunks or the coding chunks of a genome. So
okay. I'm sorry.
Yeah. So it's not exosomes. So in an eukaryote, we have about 98.8% of 98.2% of introns and about 1.8% of exons, which code for proteins. So this 1.2% of the entire exonic chunks would translate to somewhere around 34 to 38 MB region in humans. And this plays a very important role because we need to identify what are those particular mutations present in the affected individuals, where those mutations are seen in them and which are not seen across the healthy population. So,
well, if you wouldn't mind, in the DNA, you mentioned introns, you mentioned exomes. Can you talk about the various areas of DNA that occur, and then what the basic job of each is as an overview?
Yeah, certainly. So to start with, we have the nucleus as a central core of an eukaryotic cell. So we have about a trillion cells in human body, and the human genome is diploid. So there's about 3.2 billion bases of human genome for a haploid set. So which translates to about 6.4 GB of human genome in size. So the euchromatin is intercoiled or intertwined inside the nucleus, and a bit of this particular intertwined chromatin, or a bit of a DNA, is nothing but a gene. So a gene has got both introns and exons. So as I told you, in case of eukaryotes, you got a lot of intronic chunks, which is about 98.2% of the entire human genome, is making up around making up intronic chunks, which doesn't code for any proteins, but which was at some point of time believed to be junk, but it's no longer believed to be junk because they play a larger role in regulation. That's a different side of the story, but we're talking about the most important stuff, which is going to be the coding chunks. So the exonic boundaries play a very important role because it harbors a lot of mutations. Nevertheless, the mutations that are present in the non-coding regions harbors a lot of genetic variation. So you find a lot of genetic variation sitting in the non-coding RNAs, not necessarily the coding chunks. So the beauty here is, in terms of exomes, we're only talking about identifying SNPs present in these coding chunks, but we'll also talk about whether or not these SNPs, which we identify, have an influence on the SNPs present in the intronic regions or the intronic boundaries. So this is where the idea of targeted amplicon sequencing comes into picture, which means we could probably sequence an iota of exonic chunks and also a part of intronic chunks, put together. So that's where this is what we call it as a targeted sequencing or targeted amplicon sequencing. Yeah.
So you want to target. So okay. So it's possible to sequence exomes and introns. Is the whole, the entire DNA able to be sequenced, or are there parts that we still can't?
Exactly. Yeah. Certainly. Yeah. So
the whole DNA can be sequenced, exomes and introns included. Yes.
Yes. If you want to sequence the entire DNA, that's what we call it as a whole genome sequencing. But in a whole genome sequencing, the depth of coverage, although we could achieve higher depth of coverage, it would be pretty expensive. So, but things are in place now. Many researchers or many clinicians who wanted to identify mutations for their desired phenotypes are going for whole genome sequencing rather whole exome sequencing, because they believe that taking a slightly bigger depth of coverage and sequencing the whole genome would be an ideal measure, rather going for a whole exome sequencing or going for a whole transcriptome, sequencing. So, but imagine, we are paragliding or parachuting, say 10 kilometers above the Earth. We might see some gray areas, which may not really identify certain geographical regions, somewhere 10 kilometers above the sea level or 10 kilometers, from the outer space. But if we zoom in, if we come somewhere near 2 kilometers above the Earth, we might see some places which we could certainly identify. And if we can identify a certain spot where we wanted to fall in, that's where, we could zoom in further. So let's imagine we are somewhere about 800 meters or 500 meters above the Earth. And that's where, we can accurately say that, yes, this is where, we need to land, or we need to, stretch our wings and come down. So this is just like this. So higher the depth of coverage, better the visibility, and better the perchance that, we could identify the mutations. So the whole genome sequencing, when the Human Genome Project was launched, the coverage was just 2x to 4x. But thanks to the next generation sequencing these days, we could even go for 100x depth of coverage for whole genome sequencing. Albeit the fact that
What does that mean? What does that mean if if you're sequencing the whole genome? What are you missing? What does the depth of coverage mean?
Yeah, we would not be missing anything, but probably we would certainly miss the so-called bona fidelity if we are going for a lesser depth of coverage. So if the ideal would be about 25 to 30x. So we might identify SNPs. We might certainly definitely identify the mutations. We would definitely identify the structural variants. We would definitely identify the copy number variants. we would definitely identify those variants, that are coming across both intronic and as well as exonic chunks, but probably with a lesser precision. So we might need to zoom in. We might need a greater depth of coverage to check the bona fidelity of those particular SNPs or mutations that we identify. So 30x may not really be an ideal target. Probably we might go up. We might say that it's about 60x could be an ideal one, or probably 80x, or probably 100x. But again, greater the depth of coverage, greater is the cost of that particular whole genome, that needs to be sequenced. So it all depends.
I heard too that you can't leave the DNA intact to sequence it. You have to break it up into pieces, and the longer the pieces, the more accurate you are. So how does that play in?
Yeah, exactly. So that's where the basic next generation sequencing concept comes into picture. So the DNA is broken down into different fragments, small fragments, and the fragment size can be decided well in advance. So the earlier days, the fragment size used to be only 50 bases or 100 bases, but thanks to the now generation sequencing, so we have got good instruments that could read 150 bases, or the fragments that can be split into 150 bases, 200, 250, 300, these days up to 400 or even 500. And if these fragments are spliced, or if these fragments are made, they need to be identified. So we have something called adapter and linkers, ligated to these particular fragments, so that we can clamp them, and then we can identify what these particular fragments are up to. And then comes the orientation. So we can read the bases present in this DNA from left to right, or right to left, or from either ways several times. So if you are reading the DNA bases from say 5' to 3' end orientation, or one way, that is what we call it as a single end chemistry. Whereas if you read from both the ends, that is what we call it as the paired end reads. And several paired end reads make up something called mate pair chemistry, which is what we call it as MP. So mate pair reads are seldom used these days unless your lab is rich. But an ideal would be 150 base pair paired end chemistry, so which we call it as a 2 by P paired end chemistry, so which is about 2 by 150 base pair. So which means each fragment would be of size 150 bases. And then, so post library preparation, so which means you clamp the adapters and linkers, then you send these particular fragments, for sequencing through these particular machines. So this is how we know it is done. And then you map it to the reference. So usually when you map it to the reference, you need to know whether or not your sequence of interest has got any reference genome, associated with that. Otherwise, one could go for something called de novo assembly. So which means you would start right from scratch without assuming that that particular sequence has no bona fide entry into the database, or bona fide sequences associated with the database. So it doesn't have a reference. So we would start from scratch, and we'd come to know that certain sequences might be associated with so many taxonomical clades, or so many groups or genera and such as such. So this is how we know we go for sequencing, and of course the final stretch is the downstream analysis. So after you map to the reference, in case of reference mapping, we identify the mutations in case of whole genome sequencing or whole exome sequencing, and now comes a very important.
So will the fragments, at 150 base pairs or larger, will that be better sequencing, and why? Why is it better to have bigger fragments?
That's really a very good question. In fact, not necessarily. It all depends on how good the depth of coverage is. So, in case of whole exome sequencing, so if you're going for say 100x coverage, you would certainly fall short of 100x. So not necessarily the sequencing machine would cover 100x coverage of your genome. So you would probably slightly fall short of, say maybe 8x or 9x. So in principle, it would be only 90 or 91x, the coverage. Having said that, if the fragment size is bigger, you could definitely map those particular sequences to a better readability. But the precision of those particular sequences that are mapped to those particular readable references needs to be thoroughly checked. So it doesn't make any sense if the fragment length is quite big. But the last 15 minutes of my talk was largely meant for something called short read sequencing, which is like ion-terrent or Illumina based sequencing. But we could even go for larger amplicons where we could even read larger size sequences of more than, say, 1,000 bases or even 5,000 bases at one go. So that's where these PacBio, which is a well-known sequencing company that measures these particular long reads. There's also another sequencing technology called Oxford Nanopore technology, which is abbreviated for ONT. So this has become a wonderful champion for next generation sequencing these days. That is what we call it as Oxford Nanopore Technologies Minion technology. So which is a USB based stick that is connected to the laptop or your desktop, and then you can read the sequences in real time, where you simply sample the sequences in your flow cells and then measure the reads. And again, ONT has got a greater precision as well. There are several minions clubbed together. There's something called Prometheon. There's something called Gridiron technologies, which have shown some kind of success. So there's a difference between short read sequencing and long read sequencing chemistry. So in conclusion,
what's the end goal of your sequencing? You want to better identify mutations, and what does that mean, better? If so, what will you figure out that has not been figured out already?
Yeah. So I would probably choose wisely what needs to be done. So for example, if I were to identify mutations that have not yet been discovered, say for a rare diseased phenotype, I would probably start with whole exome sequencing as a pilot. And if the lab is rich, if we have got sufficient money, probably I would directly go for whole genome sequencing at a greater depth of coverage. So the ideal would be the pilot experiment would always be the whole exome sequencing, because we would always try to infer what are those mutations present in these affected population where these particular mutations are not seen in the unaffected individuals. So that's where the whole exome sequencing comes in.
What about epigenetic effects, methylation and histone action? Does that happen in the region of the exomes or the introns? I've never heard that talked about.
Yeah. So certainly that's indeed a splendid question. Seldom do we find the epigenetic changes from a whole exome sequencing, but it all depends on how the cell has nurtured itself, what kind of environmental influence this particular cell has had during its lifetime. So if you take exome, so let's imagine exome is nothing but a union of all the exonic chunks. Say if you take one gene which has say about 12 exons, so we would ideally sequence all the 12 exomes, all the 12 chunks of those particular exons, and then we could even probably target the intergenic boundaries as well. So which means these 12 exons has got about 11 introns between them. So ideally, there might be certain intergenic non-coding RNAs present in between them as well. And if we go further upstream of the whole gene at 5' untranslated regions, or even further downstream, which is 3' untranslated regions. Further 5' UTRs, you have got something called promoter, you've got open chromatin elements, you've got something called DNase hypersensitive sites, or even some kind of long non-coding RNAs. So the area between the DNase hypersensitive sites and the start site of the exon harbors a lot of methylation events. So this is where a large research is really focused on. But ideally, from a whole exome sequencing or from a clinical exome, probably it may not be a good idea to identify the methylation patterns or methylation events from a whole exome sequencing. So for this, a new technology has popped up some years ago, which we call it as a MedipSeq, which is a methylated immunoprecipitation sequencing, where the entire exons and its boundaries are sequenced and checked for any methylation patterns. So that is what we call it as a MedipSeq. So that is an ideal way to look into the methylation patterns. Nevertheless, if you're going for a higher depth of coverage, probably whole genome sequencing is also another alternative to identify the methylation patterns. Yeah.
So, okay. Are there particular conditions that you think that this new type of sequencing will show? I mean, what are the the clinical outcomes or potential outcomes for this new sequencing?
Yes, I mean, probably I'll just try to put our case study, which we have worked extensively the last 2.5-3 years. Our research has been as dealt about identifying mutations from a rare disease called pouch colon, which is congenital pouch colon. We did get some serendipitous results. So what we discovered was, or what our potential application was, to identify the mutations in a family of 16, so which is about quad. So you've got about 16 probands, which means 16 affected individuals, 16 children, their father, mother, and of course, again, unaffected sibling. So we did identify mutations that are absolutely de novo, which means those mutations that are present only in affected individuals and not seen in either mother or father. But more to that, what we observed from a whole exome sequencing was we identified a long intergenic non-coding RNA that was shown to be having an influence on one of the genes responsible for this causal to the rare disease. So this was one of our recent studies. So this is certainly a serendipitous thing. You might ask me, how come from a whole exome sequencing we could identify a long intergenic non-coding RNA which is coming from an intron? So that's where a typical library preparation was given a miss. Maybe one of the sequencing librarians who chopped off the exons, he could have probably taken certain intronic chunks as well, and invariably one of those long non-coding RNAs was sitting in those particular regions, and that was how we try to discover. So it's basically a targeted amplicon sequencing in disguise, and that is where we published, and one of our first authors, Sonal Gupta, she worked on it. And this pouch colon is certainly a very rare disease in India, in the state of Rajasthan. And our clinical collaborator, Dr. Praveen Mathur, he has done a lot of surgeries on this, and we were trying to identify a lot of mutations in these particular patients. So So our head of the department, Dr. Krishna Mohan and Dr. Praveen Mathur, they conceptualized this particular project. And these are the data sets that I analyzed as another co-principal investigator. So the take-home message here is it all depends on the kind of applications that as a scientist, one would look into. So as a researcher, our point was, of course, to identify the mutations from the exonic boundaries. But we were also looking from an ideal point of view whether or not these SNPs that we identify have got any influence on the genes or any other particular genes that play a very important role towards regulation of the disease. So this was what we asked for. The commonmost thing is from a whole exome sequencing, you'll be chopping off the entire 34 MB to 38 MB of the entire human exons, and you would identify the mutations. But in a clinical exome, probably you would only look into those genes that are specific to a diseased phenotype. So you would only look into those genes and identify the mutations only to those particular genes responsible for that particular disease. So that is where the clinical exome comes into picture. So clinical exome is invariably cost effective when compared to the whole exome or whole genome. So you would only look into a smaller component of the entire exonic chunks from the human genome. And this is where one would ideally look into. Yeah.
So are there any surprises with your level and your version of sequencing that weren't there before that you're seeing?
Certainly, this was certainly when we said it's serendipitous results. We were absolutely overwhelmed, and we were quite ecstatic, and we thought, hurrah! It's absolutely a great moment. So what we did was we of course cross-checked. We used a Smith-Waterman algorithm, which is a well-known algorithm to check the pairwise alignment, to check or to compare two sequences whether or not they're similar. So we complemented this bioinformatics strategy with an in vitro experiment using a biophysical method called macroscale thermophoresis. So and we did confirm it. Coming to the surprises, I don't think clinical exomes would identify only the SNPs coming from the exonic chunks. I mean, these are no longer surprises. These have become a reality. So it all depends on how good the sequencing librarian is, or how good a listener is, he or she is. So as a scientist or as a researcher, you may want to tell him or her that probably you want to chop off these particular chunks, these particular regions. Probably you might be interested in these particular regulatory regions of these interest. Of these interesting genes, and then he or she would ideally identify those particular sequences. So I would say in conclusion that whole exomes or clinical exomes would definitely identify long non-coding RNAs, or at least long intergenic non-coding RNAs. And it all depends on how good the libraries are prepared and how good the person is making these libraries.
What about for cancer? Is your sequencing useful in terms of identifying the particular mutations associated with various cancers?
Yeah. I mean, certainly. I mean, in fact, cancer, I believe is largely studied using the next generation sequencing technologies, albeit the fact that people have been studying familial cancer types across different disease phenotypes these days. Cancer is one commonmost area people have been identifying, but we have also ventured into identifying mutations specific to cancer prostate in Indian phenotype. So in India, cancer prostate is also uprising. So it's the third most leading cancer in India, thanks to the population-based cancer registries that we have in India. So these data sets I've been publicly available. So we could collect a lot of samples from a clinical collaborative hospital here, and we started whole exome sequencing here. And with the pilot, we have analyzed so far about 30 samples so far, and we are increasing this up to another 70 in the near future. So cancer is one of the commonmost areas, but I would say India is although it's a developed country, I would say it's developing in terms of the next generation sequencing approaches. There's a big brain drain, coming from the far west or from Europe and all those places, where the researchers or the returnees from US or Europe, they're trying to bring the kind of knowledge that they have leveraged way back in their host countries. So, but when it comes to in India, no major cancer exomes or no major, clinical exomes related to cancers have been studied. So it's just, beginning. So we are still working on it. And the government of India, of course, it has also started the Genome India project, where, it has launched about 10,000 Indian genome projects. So that is also, beginning to be studied. Yeah.
Well, very good. Well, Prash, what's the best way for people to find out more if they want to speak to you about, how you sequence, companies, individuals? How can they look you up and find out things?
That's a tricky question. With due respect to all these service providers, in a country like India, many labs do not have these next generation sequencing facilities, but they are largely dependent on the service providers. Even if they have these machines, the sequencers, they do not have the technicians. And of course, this is also the case in some parts of US or Europe as well. But what I strongly believe is that the clinicians should not be taken for a ride. They should not be taken for a ride, and they should not be fooled, saying that this is the analysis that is done say for a whole exome sequencing or a whole genome sequencing. So it's high time that the clinicians and the researchers take a big note of the happenings, especially in the field of next generation sequencing. They need to make a big know-how of next generation sequencing strategies. They need to attend certain courses. They need to collaborate. They need to go for a crisscross collaboration, understand the nitty-gritties of next generation sequencing, what's their cup of tea, and how they're looking, say, 10 years down the line towards development of their projects. So, unfortunately, from my experience in India, at least, there are very few companies who speak good science, especially in the areas of next generation sequencing. So, we should always be careful, and bioinformatics is always challenging. The next generation sequencing is always challenging. You should always reach consensus in whatever ways or whichever strategies the sequencing is done. Ultimately, what matters is how good is your bioinformatician or how good is your bioinformaticist. So that plays a very important role. So probably it's a good idea to reach consensus between two analyses. There's nothing wrong. Hey, look, you see, I've done this particular work. Would you mind, reanalyzing my stuff? So there's nothing wrong. So there's no shying away here, because what all matters is, to do good science. If one does good science, that would bring a healthy network, prevailing in the scientific community, and that would really, bring good publications, in academia. So what I always believe is the service providers should not take the clinicians, especially, for a ride, which unfortunately, from our experience, they're really taken for a ride, because poor clinicians or poor surgeons, they don't know the nitty-gritties of genetics or next generation sequencing. Whereas the researchers, the other way, they should reach consensus. They should trust their bioinformaticists. They should ensure that they reach consensus with their bioinformatics analysis. They cannot say that, yes, they have analyzed one time and they just wanted to publish it. That's not their cup of tea. They need to reach consensus. That's certainly a very, very must, I would say. So this is where we could bridge the gap between between the service providers making good next generation sequencing and the researchers identifying good bioinformaticists so as to make good publications. Yeah.
Well, very good. Well, Prash, thank you for coming and for your work and for your thoughts. I appreciate it.
Yeah. Thank you so much, Richard. You're most welcome. Yeah.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.